Okay, so in the first question, it says a scientist isolates a number of non-photosynthetic prokaryotes. The clue here is prokaryotes, and which structure would be found, okay, in a prokaryote. So if you're looking to get your points back, you need to be thinking about cell structure, because that's where we cover this. Cell structures. So here, the cell structure is where we covered this material, prokaryote versus eukaryote. Now, when you're answering this for your points, to get your points back on the test, you need to remember that you have to find out where exactly did we cover this. And of course, here you know that I'm going to let you go ahead and figure out why prokaryotes would only have cell walls and not the other three. All right, so explain that. There's actually one answer explains all three of them, but especially why is answering D for prokaryotes the worst answer. But don't forget to explain why prokaryotes don't have Golgi bodies or chloroplasts. You should be able to explain that. If you can't, you need to go back and look at the videos, or you need to see me when you get back in January before the final grades are done. All right. Again, there was a mistake with number 2. Unfortunately, there was no question there. Things got jumbled up. But 3, which prediction would be supported by the results of gel electrophoresis? Well, the answer is that cell 1 from individual A will have the identical banding pattern compared to cell 2 from individual A. In fact, all the cell samples from individual A will all have the same banding pattern, and all the cell samples from individual B will have the same banding pattern. Now, your job is to explain why that is. Any other choice, you have to explain why you chose the wrong answer. Now remember, this is remembering that when we're looking at banding patterns, they're telling you the DNA is what you're comparing. All right, let's move on to the next question. Here is this is from another version of the exam. It says a scientist isolates a number of non-photosynthetic prokaryotes. Which structure would be found in these cells? Again, same answer as before. The answer is cell walls, and it would be cell structure again. And 2, it says which means of particle transport is shown in 7-5. If you take a look at it, there's a particle, the cell surrounding it. Finally, that little black sphere is engulfed by the cell, and then the vacuole, which is sitting here, combines with the engulfed portion, and that big piece of material is made smaller, is broken up into smaller pieces of material. So what would be happening in this process? Well, the answer here is endocytosis. Now you'll have to explain where we discussed this and why the others are wrong. Okay? Anything other than endocytosis, and you have to explain why. Again, this being a different version, you're going to see the same answers. Some of the same questions, they do overlap. This is A. First of all, the answer here is B again. All the cells from cell 1, all the cells from individual A are going to be the same DNA pattern, and all this samples from individual B will have the same banding pattern. You'll have to explain why. You don't have to know what gel electrophoresis is to be able to answer this. You just have to know something about DNA. And everyone in the class has proven on other assessments that you do know. And when you answer in class, you do know. But what is it about this particular answer that makes this right, and why are the others wrong? All right, let's move on. Here, another common question between the two versions, what's going on with diffusion? Well, I'm going to ask you two questions. I'm going to ask you the same two questions I've asked over and over again in every osmosis and diffusion problem you've had since we covered osmosis and diffusion. What is it that crosses a cell membrane? What doesn't cross a cell membrane? Why is it that some things pass and some things don't? So now your job is to figure out why the answer here is water from the red blood cell will diffuse out. Remembering that according to the problem, the red blood cell has lower concentration. So if the red blood cell has lower concentration, that means that then you know that this is high and this is low. Why does water move? What do we call it when water moves? Why is water moving? Why doesn't the protein and the sugar pass by? These are the things you have to answer. You should know this. This is something you really should know. All right, moving on. Diagram models the time-lapse movement of particles. So there's these particles. Here's these. Obviously, this is high. It's going to move towards the low. The particles move. Here, the particles are equally distributed across the membrane. Obviously, particles are going to keep moving back and forth. So now let's go ahead and write that, and you're going to choose the ones that are correct. Remembering that when we did this, we changed this and this to A and B. All right, so what answers would be the correct answer? Well, the particles crossing membrane do not have a charge. Well, that makes sense because what crosses the membrane? Things that don't have a charge, things that are neutral, right? Non-polar materials. Oh, I'm sorry. This one, A. So A is correct. The movement across the membrane requires energy from ATP. Well, the question that you have to answer is, if you have a high and you have a low and things are moving from high to low, that's the direction of the movement, right? Is from high to low until it finally reaches equilibrium. Then do you need any extra energy? Well, the answer is no. All right, so C, the movement across the membrane requires energy from glucose. Well, again, same answer, so that's not an option. The movement across the membrane does not require added energy. Well, that one's right. Remembering that this is what the state of Ohio requires you to know. All right, and you should know this. We've talked about this forever. Particles crossing membrane could be proteins or carbohydrates. Well, that's a no. That's not possible. All right, and this one can't be, and this one can't be. Why can't they be? That's what you have to do to get your points for this question. The particles crossing the membrane could be water or oxygen. Yes, because water does cross through the membrane and oxygen is neutral, right? Is small and non-polar. Water's polar, but it's small, and in small amounts it does cross. So A is an answer. Well, A, if you chose A, the first A, it's already filled in on your Scantron. Choosing A again, you didn't choose, and it would still be filled in. You don't have to do anything else. So you would have on your Scantron A filled in and D filled in right now. Now let's look at B. The particles, the second B. The particles crossing a membrane have a strong positive or negative charge. Well, again, the answer there is no. Again, your job is to answer why is it no and explain what rule we're following when you apply it here. So again, what crosses a membrane? How does it move? Why does it move? And do you need energy? All right, moving on. Here is an enzyme. Here's a photosynthesis. We know enzymes are involved in photosynthesis. We talked about it in depth. The light, the photosystem 2, photosystem 1, then later the Calvin cycle. So we talked about photosynthesis in general. here's a chart of photosynthesis. As we look at the chart, ask yourself what's going on as photosynthesis starts at 0. You have probably what? You have 2 gases you have to worry yourself about, right? CO2 and oxygen gas. Those are the 2 gases. And so where those are, if you read the question, it says which statement is supported by the graph. So we have to worry about these 2 gases. What's going to happen to CO2 as you move through photosynthesis? Are you producing CO2 or are you using CO2? Are you producing oxygen? Are you using oxygen? As you increase photosynthesis, are you producing more oxygen? Are you using up more CO2? That's what you have to think about. And all you have to really do is write out the equation that you've been taught, that you've learned, that you've been tested on. CO2, right? Plus what? Water yields C6H12O6 plus what? Oxygen gas. All right, so you should know that. You should know what's going on with that. So forget the mess, but I'm doing this while I'm driving home, and I want to make sure that you get this as soon as you can get this as soon as you can, so you can get your work done and start your vacation. All right, which statement is supported by the data in the graph? The rate of oxygen production is the lowest in section M. Well, are you producing oxygen if you're doing photosynthesis, or are you using oxygen? Look at the equation. Go back and review your notes if you don't know the answer to that. So the rate of oxygen production is lowest in M. That would be no. The answer to that is no. There's a reason for that. Explain it. The rate of oxygen production is greatest in M. That answer is correct. The production of water molecules is greatest in L. That would be wrong. Number of carbon dioxide molecules in the beaker is greatest at K. That would be wrong. Where is CO2 the greatest if it's not at K? Why is it greatest at wherever it is? And why is it not greatest at K? Those are the questions you have to answer. Which statement explains the effect of temperature on the rate of photosynthesis? Well, oxygen molecules break. Now, what are we talking about? We're talking about the graph that we just previously looked at. You can rewind it and look at it. It went up, and then it crashed at a certain temperature. I think it was around 25 degrees Celsius. So why would any reactions happening that's driven by enzymes, why would it then collapse at a certain degree? So let's take a look at what these answers are. The oxygen molecules break down at temperature above 25 degrees. That doesn't make sense. Oxygen molecules do not. If that were true, it's often above 25 degrees, especially in the desert in the summer. There would be no oxygen in the desert, right? That wouldn't make sense. The rate of photosynthesis is unaffected by the temperature of the water. Well, we know that's not true because the line moves up and down. So again, reading the graph. But I'm not going to tell you anymore. Take a look at it and come up with your answer explaining why that's neither one of those are true. The number of chloroplasts in a cell decreases with the temperature. That's not true. The enzymes involved with plant photosynthesis are disrupted by temperatures above 25 degrees. That is the best answer, and now you have to explain why. And to reference that, I would tell you look at enzymes or discussion on enzyme action. What happens to enzymes? I'll remind you of 2 words: denature and renature. All right. The idea is that all living things are composed of cells, comes from other cells, defines what? That would be cell theory. All right. It's not any of these others. The answer is cell theory, and I told you should look at the other questions. You'll see there's another question that goes through some of the components of cell theory. You would expect a cell with an extensive Golgi apparatus to do what? Well, the answer is to secrete a lot of material. Now, how do you answer that? How do I know that? Let me ask you this. What does a Golgi body do? All right. And then a secondary question is, what makes ATP? And what would a Golgi body have anything to do with that? If something's really active, what do you need a lot of? Probably ATP. So what would you need a lot of? The thing that makes ATP. Does the Golgi body make ATP? Perform photosynthesis. Does the Golgi body do photosynthesis, or is that a chloroplast? And store large quantities of food. Again, what is the function of each of your organelles? There should be no question that anybody got these questions wrong. I'm surprised about it. The function of the nucleus is what? Well, the manufacture polypeptides. What makes proteins? What makes polypeptides? This is a transcription translation question, right? People were like, where are the transcription translation questions? Right here. What makes polypeptides? Not the nucleolus, right? Help manufacture ribosomes? That's correct. Why? Because the nucleolus, I said nucleus, I believe, a minute ago, but the nucleolus makes the RNA for ribosomes. So I gave you that answer, why that's correct. Now explain why the rest of these are wrong. By the way, remembering that chromatin is DNA. All right, it's a form of DNA. A cell has mitochondria, ribosomes, smooth ER, and other parts. Based on this information, it could not be what? I hope everyone understands it can't be a prokaryote. It cannot be a bacteria. It cannot be a bacteria. These are things it could be, because it has all those things. And of course, it can't be this one as well. The blank of a mitochondria is an adaptation that increases the surface area. So it has increases surface. It has to have a lot of membranes. What is this thing that increases surface area? That would be the cristae. All right. The rest of these are wrong. Why? Again, what is it? Just structure. Structure and function. What do these structures do? Come on. The stroma is the blank. The stroma is the area between the inner and outer membrane of the chloroplast. The rest of these are wrong. What is the space in between the inner and outer membrane of the mitochondria if it's not this? The others are wrong. Why? Cilia differ from flagella. Well, that's easy. The answer is this one. And I told you the others we haven't covered. We haven't discussed. Don't have to worry about it. This one's easy. Why? Because cilia are those little the little projections that move, a lot of them, and the flagella is only one or a couple that are longer and they swirl around. They look like a tail, like that on the sperm. Organelles involved in energy conversion are what? Should have been easy. Should have been mitochondria and chloroplast. That's ATP production. It's not any of these. Why? What are the function of each of those organelles? So which of the following consists of prokaryotic cells? Now again, as I said earlier, these questions I really feel are pretty straightforward. What are prokaryotic cells? Come on, guys. Are they plants and animals? No, they're not. Are they plants? Are they plants at all? Absolutely not. And they're not fungi. So that's not true. Are they animals? You just said not plants and animals. You should know that. You should know that plants and animals are eukaryotes. Why are they eukaryotes, though? What makes them eukaryotes? And that's a fundamental question that everyone should know. Plants, bacteria, plants again. There's plants again. So it's not that one. So I think maybe some people got confused because some of the answer choices had bacteria and archaea repeated. And someone asked me, well, what is archaea? Well, you should know archaea is a form of prokaryote. It's a type of for for discussion purposes, it's a type of bacteria. It's a type of prokaryote, something without a nucleus, right? Very, very, very disappointing. Anyone got that one wrong. So the answer is bacteria and archaea. Again, go back in the notes. We classified organisms. When we did our review for the final, we looked over what are the different kinds of cell types. We have fungi, we have animals, we have bacteria. We said we have archaea. We said we have different kinds of structures in each of these. What are they? We said repeatedly throughout the entire semester that fungi and plants have cell walls. We said fungi cell walls of plants are made of cellulose. We said that the cell walls of bacteria and that of fungi are made of different materials. We talked about what they're actually made of, but we didn't ask any of those complicated questions here. The only question you were asked here is which one of these could be prokaryotes, and there's only one possible answer. Now you go through and explain why this one's right and the rest of those are wrong. Okay, if you got it wrong, if you got this one wrong, right? All right, so all the following statements would be supported by cell theory, except now this one's related to the other cell theory question. When you're taking a test, you come up to a question, you don't remember what cell theory is. Well, all of these are right except for one. All living things are made of cells. That is part of cell theory. All cells come from other cells. That is correct. The cell is a basic functional unit of an organism. That is correct. Cells reproduce to form additional cells. Also correct. Very similar to the characteristics of life, right? So because those are correct, right, and the question says except, then that makes all of those wrong. So we don't choose these. So again, maybe that's why some people got this one wrong when you're talking about test-taking skills. The word except is there, right? So the cells grow to unlimited size? No. That's surface to area volume. When we went through and did that activity and calculated the surface area to volume ratios, and we said cells can't get bigger than this. That was part of the work you did in preparing for this unit of cell structure. Which of the following statements best describes an organism's attempt to maintain cellular homeostasis? Homeostasis. The example we used in our test review was the thermostat on the wall. We talked about temperature, right? Keeping how we have to keep you within a specific range. You can look at the video for the week of our test review, and you'll see this is emphasized. We talked about temperature. We talked about pH. We talked about keeping a salt balance. All that. All right. So what's which best describes, not that any of these don't describe it, but which best describes cellular homeostasis, right? So it's homeostasis at the cellular level. A flowering plant bends towards the light. Maybe, but it doesn't sound like at the cellular level, is it? It's not the cellular level, right? A paramecium's contractile vacuole expels excess water from the cell. Well, think about what that means. Paramecium, a single-celled organism, has a vacuole inside, a contractile vacuole, that holds water. So why would it put water out? Because too much water is coming in. You're out of balance, right? You have too much water coming in. So that means that this contractile vacuole has to squeeze or close and kick water out to balance the water coming in. Otherwise, the cell will explode. This is a problem for freshwater organisms. We talked about this when we talked about a contractile vacuole and what it does. So, in any case, that's the right answer. Let's keep going to make sure there's not a better one, though. A wren lays 3 eggs in its nest. Well, is that really homeostasis? In some odd way, it might be, but it doesn't really speak to the cellular homeostasis. A pup growls when a litter mate attempts to take its bone. Again, not cellular. And earthworm's DNA is replicated. Again, really, what does that have to do with with the idea of homeostasis, of keeping something within a range, within not too high, that Goldilocks idea where you have, you have a temperature that's not too high or not too low, or a concentration that's not too high or not too low. So go back and try to figure out what the purpose of that question was about, and why those are wrong and why they were right. Which of the following structures would be used to identify a prokaryotic cell? I guess if you don't know the difference between prokaryotic cell and eukaryotic cell, you're in trouble. You're in trouble in this exam anyways. And I don't understand why you wouldn't know that, but there it is. Prokaryotic cell. Well, what are the prokaryotic cells? That's for you to look up. You should know, but go ahead and look it up if you don't. Well, do prokaryotic cells have flagella? Do prokaryotic cells have chloroplasts? No, they don't. Do prokaryotic cells have smooth ER or rough ER? No, they don't. Do they have mitochondria, Golgi bodies, or lysosomes? No, they don't. Do cell membranes? Do they have a nucleus? Certainly, they don't have a nucleus. So the only possible answer is this: do they have cell membranes, ribosomes, and chromosomes? Yes. So again, why do they have these and not these? It goes back to the definition of prokaryotic cell. Look it up. Write it down. You should know this. Plant cells contain both cell wall and cell membrane. What is the composition of the cell wall? And where is it located in relation to the cell membrane? Well, here, this one, you might have gotten this one wrong. I'm not sure why you would, but it's easy. It seems to me that if there's anything among these questions that would make it easy for you to get confused, it might be the idea that the cell wall is outside of the cell. But you've done osmosis and diffusion questions where you've seen where we've drawn a plant cell that's going through plasmolysis, and the membrane looks something like this, and it was shrinking, and it was stuck, and it was attached to the cell wall, and the cell was shrinking. And you also had one where the cell was swollen. So we've drawn these out in class. You've been tested out in the last unit where the cell was swollen when it was in a hypotonic solution, and when it was in a hypertonic solution, when it was higher concentration salt outside, what you saw is the cell going through plasmolysis or shrinking, and the other one was swollen. And you had this kind of drawing. In fact, you had this on your osmosis diffusion quiz or exam. So there should be no question that the cell wall, which is represented by the blue line, is on the outside of the cell. But again, go back and identify the Y's and what fours and where this material was discussed. So where is it located in the cell? Really, it's outside the cell. So and it's made of cellulose. So it's cellulose and outside the cell. This one's wrong because it says inside. This one's wrong because it says glucose. This one's and it says inside the cell for both reasons. It says glucose, and that's why the C is wrong as well. So there, I gave you a lot of your answers there. Here you have a graph. I mean, I think it's pretty obvious that this area here has a lot of absorption going on. And how do I know that? You can read the chart. It says percent absorption, a lot of absorption. The only light that's going to be used for photosynthesis is the light that's absorbed. We've gone over this. We talked about why plants are green and what it means for us. We talked about wavelengths and frequencies. And we thoroughly went through this. And then, of course, it absorbs a lot of light there, absorbs a lot of light there. Just eyeballing it. That's what I would say. So it says the graph indicates it says the graph indicates that the energy used in photosynthesis is most likely obtained from which regions of the spectrum? Well, violet and blue, violet, blue, but not green. Why not green? Well, that's for you to figure out. You should know that. So you know that's why that's not correct. What about yellow and orange, red? Well, yellow's here. Is that really most likely obtained? The energy is most likely obtained from yellow and orange, red. So yellow, orange, red, okay. Yellow, orange, red, and blue, violet. Orange, or blue, violet, and orange, red. So what do you think? I think that one's probably the best. That one's probably the best. And so that would mean that this can't be the answer. And green, again with the green, you should know it's not, definitely not green. All right, moving on. If the test tube below were allowed to stand at room temperature, so you have a test tube, it's airtight. Here's some yeast and glucose. So the yeast have food. They have a little bit of oxygen here. So they're going to start doing cellular respiration. But what's going to happen as the oxygen runs out? As the yeast run out of oxygen, what are they going to do? Well, if you've been paying attention, you'd know they do fermentation. And why do I say fermentation? Because it says after several hours. Well, after several hours, there's going to be a lot of yeast in here. And they're going to be using a lot of oxygen. They're going to run out of oxygen. What's the only possible answer is fermentation. Not oxidative respiration, right? Why not oxidative? Because they'd run out of oxygen. That's the point. So it's not oxidative respiration. Is it photosynthesis? Well, yeast don't do photosynthesis. Nobody should have chosen that. If you did, explain to me why you would choose photosynthesis. And evolution is not part of this discussion at all. So, all right, let's go on. The yeast did not evolve in the several hours. That doesn't make sense. Moving on. All right. So here you have another setup. Let's take a look at it carefully. You have a plant. It's underwater. It's capturing the gas bubbles that are coming up. Where are those gas bubbles coming from? They're coming from the plant. There's light. There's nutrients in the water. There's water. What's it doing? What do plants do? Everyone should know this. Plants do photosynthesis. So what are they producing if they're doing photosynthesis? They're producing oxygen. Right? And they're asking what gas is released. Certainly it's not releasing carbon dioxide. What's it doing to carbon dioxide? What do plants do to carbon dioxide? They fix them. Remember that enzyme Rubisco? They fix them. It captures the CO2, reduces the CO2. It goes back to that graph question you saw earlier. So CO2 should be dropping, not increasing. And oxygen should be increasing. Where's it getting the CO2? It's getting it from the water. There's carbon dioxide dissolved in the water. The CO2 is coming out of there. Where's the oxygen coming from that's in these bubbles? From the breaking up that water, from taking water and breaking it up. Which of the following diagrams represents the correct cycling of gases between photosynthesis and respiration? This is, again, the third question. Where's oxygen going? Where's it coming from? Where's CO2 going? These were literally questions on your photosynthesis respiration exam. I said, if you got it wrong when we went over the exam, make sure you know it for the final. We went over this again for the final review. Respiration and photosynthesis. Respiration is producing oxygen. Using oxygen, and photosynthesis is producing it. So let's take a look at these. Whatever goes into one comes out of the whatever goes out of one comes into the other. Whatever comes out of the other goes into the first. This is something that was on your last quiz as well. So here's respiration. What is respiration doing? Is it taking in CO2? You respire. You breathe. Are you breathing in CO2? So if you chose A, I wonder why. Please explain yourself. All right, so it's not A. And explain why it's not A. You can do that, I think. I believe that you can anyways. C says Oxygen goes into photosynthesis, hydrogen ions come out. Is that true? I hope you say no, especially since I just repeated several times with the process. This is like the fourth question that has to do with photosynthesis or respiration. Something you've already been tested on, you've already been explained. Thought it'd be easy. So B, respiration photosynthesis. Does respiration take in hydrogen and put out CO2? Well, it does put out CO2. Photosynthesis does take in CO2, but when you breathe in, do you breathe in hydrogen gas? Is that what you do? Well, I hope you said no. And finally, oxygen gas goes out of photosynthesis, cell respiration oxygen goes in. It breathes out carbon dioxide and it goes into photosynthesis. Why? That is the answer. Now you have to explain why that's correct and why the others are wrong. You should be able to do that. If you can't, you need to be able to do that before January. Get it done and turn it in. All right, photosynthesis would do phosphorylation of ADP. What do we do with ADP? ADP, what do we do with ADP? Well, we add phosphate to it. That's what we call phosphorylation. And that makes ATP. That's what everyone should know by now. You should know that. That's what we do. We went over it. You did a project on it. You did a lab on it. You had a whole cell energetics packet on it that everybody said they did. No one copied. Everybody did it. Okay. You took an exam on it. Okay. So why don't you know what we're doing with ADP? We're doing what? We're adding phosphate. What's that doing? Is that putting energy in or is that taking energy out? Well, we know when we take ATP and it goes to ADP plus phosphate, we know that that's releasing energy. We've done that 100 times. That's releasing energy. So what is it? Knowing the first law of thermodynamics, we can't create or destroy energy. If taking ATP, making it ADP releases energy, what do we have to do to make ATP? We have to put energy in. And as I said in class for you, the answer would be, well, I didn't say the answer, but I said you would be putting energy in. So this would be absorbing energy. It would not be releasing it. This is not a dehydration reaction, and it's not catabolism because phosphorylation means you're adding phosphate. So that's anabolism, right? Where does electron transport and cellular respiration happen in the animal? It's the electron transport chain. Where does the electron transport chain happen? Well, hopefully everybody knows it's in the mitochondria. I mean, not everybody, obviously, but most of you, I hope, know it's in the mitochondria. Oxygen is vital to the process of aerobic cellular respiration. It allows aerobic organisms to derive a great deal more energy from glucose than anaerobic organisms are able to do. So aerobic, let's think about what aerobic means. Well, the clue is here. It says deal more than anaerobic, right? We know that there's a difference between anaerobic and aerobic. Aerobic exercise, meaning you're using a lot of oxygen. What's the vital role of oxygen? See, they told you. You pretty much knew that aerobic meant oxygen. The question pretty much explained it to you if you read it and used context clues. It is the reactant of glycolysis necessary to break down glucose into pyruvic acid. That is not correct. Oxygen is not used. Glycolysis is anaerobic. As most of you, I graded your quizzes. I read your answers on your essays for your quizzes, and the vast majority of you know that glycolysis is anaerobic. It does not use oxygen. Oxygen is a waste product of the Krebs cycle. Oxygen is a waste product of the Krebs cycle. Well, respiration uses oxygen. I've had like 5 questions on that. The Krebs cycle is part of of cellular respiration. You know this. I know you know this because you answered it in your essays correctly. You told me the 3 steps of cellular respiration: glycolysis, Krebs cycle, electron transport chain. Krebs cycle electron transport chain happen in the mitochondria, glycolysis in the cytoplasm. You knew this. So it's neither one of those two. Oxygen is required to build acetyl-CoA molecule. No, it's not. It's not used in the Krebs cycle. So it's not going to be involving, it's not going to involve acetyl-CoA. What does oxygen do? Well, I want to say probably for the 20th time throughout the semester, in probably the 5th different iteration of this same statement. Oxygen is the final electron acceptor in the electron transport chain. That we discussed a game of hot potato as an analogy. We looked at the electron transport chain in detail. We said, look, all you really need to know is this. We did it over the review for the final. We went over it again. Specifically, these were all mentioned in the review week before the final. The first plants to grow after a fire are called what? Well, the first plants, first of all, what is a pioneer? What does pioneer mean? They're the first. That's what pioneer means, is first. Right? What were the pioneers? The people that went to the West and settled the Europeans that settled the West. We called them pioneers. Why? Because they're the first Europeans that go into the West. So pioneers, pioneer species. And we talked about it. You can go back and review the videos on it. And we did it in August. We did it again during finals week. I did it right after the Monday exam. I went through and explained it in the short video that I sent you on Monday. So it's certainly not the answer is pioneer species. It's not climax community. It's not succession community. It's not seeds. And you can explain why. Moving on. Ecology of succession refers to what? One species replacing another gradually over time. You can explain why it's not natural selection, survival of the fittest, or balances of power. And that would just be by definition. You don't have to go through and explain what balances of power is or anything like that. That's just straightforwardly one definition. By definition, its succession is one species gradually replacing another, being replaced by another. A pioneer species, such as blank, are a combination of algae and fungus and grow in a rock. Now this one, maybe you didn't know what legumes were. That's fair. You never heard of legumes. Maybe you never, you know mushrooms. You know mushrooms are fungi. All right. But this tells you a combination of algae and fungi. Actually, to two of the two groups that took this test, I explained before the exam what a lichen is, a combination of algae and fungi. I said they're lichens. Those are what lichens are. And they grow on rock. They grow on bare rock. That's why it's a pioneer species. What does it do? It breaks down the rock. It helps make soil over a long period of time. So mistletoe is wrong. Mushrooms are wrong. Moving on. A type of succession that occurs on abandoned farmland is called secondary succession. Primary succession is when there's no soil. Any other kind of succession is going to be secondary succession. So primary succession is when there's no soil, like on bare rock, like when a volcano makes new land. You have to make soil, and that's why you need pioneer species. It's not climax community, it's not pioneer species. What type of vegetation would you expect to find in abandoned farmland after 150 years? Would you expect that there still be grasses growing or little bushes after 150 years? Well, I would hope that you'd say trees. I hope you say that after 150 years you have enough soil and assuming there's water, you'd expect to see some trees. There's some arguments you can make, but bottom line, the best answer is trees. Natural disasters such as flooding and tornadoes are linked to what? To secondary succession. Secondary succession. Primary succession means there's no soil. So when you have a forest, if a tornado or a fire or flood comes along and kills all the plants, leaving them to rot there, what happens to those rotting plants? They turn back into the soil. So now that soil is really rich and ready to start growing. You have soil, so it's secondary. Primary means all you have is rocks. You don't have soil. It's not old field, and it's not ecosystem. You have to explain those. All right. The final stable community that is made up of a mature hardwood tree is called the climax community. That's what it means. Climax means you're at the end. You have all the mature trees that take the longest to grow, the most amount of soil. Again, I refer you back to the videos in August or back in the review that I sent out on Monday. All right. 35. Blank succession is more likely to take thousands of years before trees are seen. What kind of succession takes thousands of years? The kind that requires you to make soil, because soil needs to break rocks down. How long does it take to break down rock? Biochemically, a long time. So what kind would do that? What kind of succession requires you to make soil? Primary succession. Secondary succession already has soil, so it won't take thousands of years. Ecological is wrong, Oldfield's wrong. 37 to 38 has you look at this. Here's a regular activation energy. That's that hill we went over and talked about. A ball rolling down the hill. Do you need energy to roll a ball back up the hill? We had all kinds for months. We talked about this. Here's the activation energy at the tall hill. Here's the shorter hill. And this is the one with the enzyme. So this is the enzyme. What does an enzyme do? Reduces the activation energy. Go back and review those videos we talked about in class. There's evidence posted on YouTube that we clearly discussed this. Repeatedly, you were told it was going to be on your test. It was on your test. You were told that it was going to be on the final. We went over it during final review. Activation energy is reduced by an enzyme, which makes it easier for the chemical reaction to go on. So which one of these represents the activation without an enzyme? It's not A, because A is the baseline energy. It's not E, because that's what's at the product. These are reactants. These are products. The progress of the chemical reaction. So it's either B, this one, or it's C, this one. Remembering that what we're doing here is we're drawing a dotted line across. So here's it. Let me do that again. Here's C, and there's B, right? There's B. So which is it? B is the one with the enzyme. Is B going to go faster, or is C going to go faster? Well, B's a smaller hill. It's going to go faster. It's easier to get over that hill. So the answer for without an enzyme would have to be the answer with which one B or C is going to be without an enzyme. That would be C. Represents the activation energy with an enzyme. That would be B. All right. And that's it for one version of the test. Now you'll see the second version, you have these. Oh, well, let's go. We still have a couple more problems here. If a gene had the following sequence, ATG, CCC, GTA, AAC, CTG, GGC, CCT. Assume the amino acid in the middle was the middle of the gene, so you don't need an AUG to start. And it has no need to start, so you don't need to start codon. What amino acids would this sequence code for? Well, let's take a look at it. ATG would be what would be on the mRNA if the DNA says ATG? It would be U, A, C. UAC is TYR. Well, that means it's only one of these three. Okay, so the first one's TYR. Fine. The second one, CCC, what would it be on the mRNA? It would be GGG, right? So G, G, and G would be GLY. So now it's one of these two. It's either it can't be any of the others. It has to be one of these two because it's TYR then GLY. Well, what's the next one? The next one is GTA, which should be CAU. CAU. CAU is histidine. So now they're both in the running. It's either C or D. Still C or D. What's the next one? AAC, which would be what? UUG. UUG. UUG is leucine. So that's it. We have a winner. It's C. Now, why is it not the other three? You should be able to do this. This is something you were told you were going to be able to do. I couldn't be more clear that this was going to be on the test. We did these problems in class. You did practice problems. You were given practice tests on this. I don't know what else to say. Which is the path that a protein takes when it's being secreted? There's that word secreted again. It means put out of the cell. Well, the first step, again, going through transcription and translation, first thing it's done is you transcribe the mRNA polymerase in the nucleus, right? That does not happen in the cytoplasm. Translation is not first. Translation is not first, and translation does not happen with RNA polymerase. Translation is what happens with the ribosome. Next thing, transcription of, by RNA polymerase in the nucleus, translation by a ribosome in the cytoplasm or endoplasmic reticulum. Then it goes into a vesicle, moves over to the Golgi body, has to go to the Golgi body, which then puts it in a vesicle, then takes it out of the cell. This was literally given to you on the last test, and we went over it and explained it. And we talked about it again during our review week. Vesicle transcription in the nucleus. Vesicle transcription in the nucleus, not correct. Golgi, it starts at the Golgi? No. Now you can explain why not those and why B is correct. All right, moving on. Now this is the same, these questions were the same in both versions, so we're good with that. Again, the same. Again, the same in both versions. And this one, these were all the same in both versions again. Here again, the same in both versions of the test. Here, both the same in both versions again. Here, I'm reading to see if these were the same. Yep, they were the same. And here, again, the same. Same questions again. Same questions. Same questions. Same questions. And same questions. All right. So that's it. I hope this helps. Please make sure you do what we had discussed, what you read in your Jupyter grades. I'm posting it now, and then I'll email it to you as soon as it's done processing. Have a good day. Remember, you have an opportunity to get 100% or close to 100%, get an A on your final by processing all the wrong answers you got on that final. Remember that you have to get any of the missing work in. I wish you would have done your final review before the final. I really wish you would have done that and really asked questions and really involved yourself. You cannot learn this stuff by just quickly memorizing it for a second and then expect you're going to remember it a month later. It just won't happen. So think on it, study, do your work, catch up. All right, have a good one.